Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we're going to be continuing our discussion on enzymes, and we're going to be talking about classification and binding models. So there are 6 major classes of enzymes, and a mnemonic you can use to remember them is low hot. So you have ligase, isomerase, lyase, which makes up the low, and then you have hydrolase, oxidoreductase, and transferase, which makes up the hot. So let's start. Ligases. The function of the ligases is to catalyze the joining of 2 large molecules, and this is usually with the input of energy from ATP or a similar molecule. Their mechanism is that they often form carbon-carbon, carbon-oxygen, carbon-sulfur, or carbon-nitrogen bonds. A biological example is DNA ligase. It joins Okazaki fragments during DNA replication by forming a phosphodiester bond using energy from ATP. So now you want to think of ligases as linkers. Think that they make new bonds and they require energy to do it. Ligases are most likely to be encountered in nucleic acid synthesis and repair. Now let's talk about isomerases. These rearrange atoms within a molecule to form an isomer. This includes structural isomers and stereoisomers. They catalyze intramolecular shifts of groups or atoms without adding or removing atoms. So an example would be a phosphoglucose isomerase, which converts glucose 6-phosphate into fructose 6-phosphate in glycolysis. These are common in metabolic pathways, and they often make molecules more reactive for the next step. Some isomerases can also be classified as oxidoreductases, transferases, or lyases based on mechanism. Remember that isomerases catalyze reactions between stereoisomers as well as constitutional isomers. So lyases. These break chemical bonds by means other than hydrolysis or oxidation. They often form a new double bond or ring structure, and they catalyze the cleavage of a single molecule into 2 products. And since most enzymes can catalyze the reverse reactions, the synthesis of 2 molecules into 1 can also be done by a lyase. They frequently involve elimination reactions. An example is aldolase. This cleaves fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and DHAP in glycolysis. And a tip is that lyases cleave without using water. This is a key difference from hydrolases, which brings us to hydrolases. Now hydrolases break chemical bonds by adding water, which is hydrolysis. They often act on esters, amides, glycosidic bonds, and peptide bonds, and they're named only for their substrate. So common hydrolases are phosphatase, proteases, peptidases, nucleases, or lipases. Now a lipase breaks down lipids, a protease cleaves peptide bonds in proteins, peptidases break down proteins, nucleases break down nucleic acid, and phosphatases cleave phosphate groups from another molecule. So if you know that you are breaking some sort of chemical bond and the enzyme has a different name that you don't recognize, it's not one of the 6 or the other 5 classifications, then it's a good chance it's a hydrolase. Moving on to oxidoreductases. These catalyze oxidation reduction reactions, or redox reactions, transferring electrons or hydrogen atoms from one molecule to another. They usually involve cofactors that act as an electron carrier like NAD+, NADP+, or FAD. So an example is lactate dehydrogenase. This converts pyruvate to lactate, reducing NADH to NAD+ in the process. Now you always want to identify the electron donor, which is the reductant, and the acceptor, which is the oxidant. Enzymes with dehydrogenase or reductase in their name are usually oxidoreductases, and enzymes in which oxygen is the final electron acceptor often includes oxidase in their names. Finally, we have transferases. These catalyze the transfer of a functional group, like a phosphate, amino, or methyl, from one molecule, which is the donor, to another, which is the acceptor. These do not require ATP input unless the ATP itself is being transferred. Now, most transferases have straightforward names, but remember that kinases are also transferases. A kinase transfers a phosphate group, often from ATP to substrates. An example of this would be hexokinase. You also have transaminases. These transfer amino groups in amino acid metabolism. And so a tip for transferases is that they are often involved in signal transduction and metabolism. Moving on, so for the MCAT, enzyme substrate binding is best understood through the lock and key and induced fit models. These describe how enzymes recognize and interact with substrates to catalyze reactions. So the lock and key model suggests that the enzyme's active site has a specific shape that perfectly matches the substrate. This emphasizes specificity. Only substrates with the correct shape can bind. However, this model assumes the enzyme is rigid, which limits its explanation of how enzymes work in more flexible or dynamic conditions. We also have the induced fit model. This offers a more accurate view. In this model, the active site is not perfect fit at first. Instead, as the substrate approaches, the enzyme undergoes a conformational change that molds the active site to better fit the substrate. This explains how enzymes can accommodate substrates with similar structures, and it also accounts for the increased binding strength and catalytic efficiency after the substrate begins to interact with the enzyme. Now imagine that enzyme is a foam stress ball and the substrate is a frustrated MCAT student's hand. What's a desired interaction? What wants to happen? Well, the student wants to release some stress and relax. And as the hand squeezes the ball, both change conformation. The ball is no longer spherical, and the hand is no longer flat because they adjust to fit each other. In this case, the substrate, the student, has induced a change in the shape of the enzyme, which is a stress ball. This interaction requires energy, and therefore this part of the reaction is endergonic. But once the student lets go of the stress ball, we have our desired product, a relaxed student. Letting go of the stress ball is pretty easy and it doesn't require energy, so this part of the reaction is exergonic. And just like enzymes, foam stress balls return to their original shape once their crushers or the substrates let go of them. I hope you now understand that the shape of the active site becomes truly complementary only after the substrate begins binding to the enzyme. And similarly, a substrate of the wrong type will not cause the appropriate conformational shift in the enzyme. Now, that's all I have for today, but next time we'll talk about enzyme kinetics.